So, hello students, and welcome to another live TA session class for the course Environmental Biotechnology. So, we are currently in week 7, and today is 6th, which is a Saturday, 3 to 5 p.m. We will be having this class. Live TA interactive session where I am the teaching assistant Ashutosh Behra, and you guys can interact with me while solving these questions which I am going to post in the class today. And these sessions will help you, one, clear your doubts, two, also give you an example or practice of your problem set assignment which you get for your tests, as well as for the certification, course certification, right? So today we'll be dealing with some hand-picked questions, 10 questions from lecture week 7, from all the lectures that have been covered in lecture week 7. This course is taught by Professor Pinaki sir, and my name is Ashutosh Behra. I'm a PhD PMRF scholar from IIT Kanpur who will be taking these live sessions. Okay, there are other two TAs which are also trying to teach and clear doubts for this course in a similar manner. Hope you attend all these classes and take the maximum benefit out of these TA sessions. So, for your information, these classes will also be recorded. The recordings will be uploaded on YouTube, and the video and meeting link will be shared to you by the NPTEL authorities. And the material or the questions and answers and brief explanation that I'm going to provide is also a PowerPoint based material that will also be shared to you in a Google Drive folder for all those students who are attending the live classes. Hence, it is important for everyone to attend these classes, clear your doubts, and sail through this course smoothly. Okay, with that much of an introduction, it's already time to start our class. So let me just share my screen and we can start the class. Okay, so this course and live TA session is brought to you by NPTEL, and this course is Environmental Biotechnology taught by Professor Pinaki sir from IIT Kharagpur. And my name is Ashutosh Behra. I'm a PMRF PhD scholar at IIT Kanpur doing the TA session for this course. Okay, I hope everyone can see my screen and hear me talk. Just let me know in the chat box. Yeah, cool. Thank you. So, this is a live session as well as a recorded session, which the recordings will be available on YouTube after this class ends. And this is going to be the format of this class. So I have prepared 10 multiple choice questions which are a collection from previous assignments and papers for this course corresponding to lecture week 7. Today we are dealing with lecture week 7. So if you have been through all the lectures of Professor Pinaki sir in the week 7, you will be able to answer these questions. And I will pose these questions in a sequential manner from question 1 to question 10. These are multiple choice questions, the exact same type as you get in your assignment and quizzes. Only one option is correct, and I will pose this question and then ask you students to give me the correct answer. With your knowledge you can tell me what is the correct answer. This is a communicative class, but I would request everyone to please turn off your video and mic during this whole session for smooth conduction of this session. You can talk to me and chat with me in the chat box. In the comment box, you can post your answers in the comment box, which I will be following regularly to collect options and answers for every question. Okay, So once I post the question, I will read it aloud and you guys have to look at the options, tell me what is the correct answer. I will reveal what is the correct answer and then we will understand the logic and rationale behind why the correct answer is the correct answer. Then we will also clear the concepts related to this question and then move ahead to the next question. I hope this format is clear to everyone. The old students and students who have been regularly attending this class are all aware of this, but this general set of instructions was for all the new students. And anyone who is requesting to join this call will be allowed to do so. So if you have any friends or anyone who is joining late, please feel free to tell them that the class is open for everyone. Okay, with that, let us start with the first question of this session. Question number 1. Question number 1. Which of these habitats has the lowest fraction of culturable microorganisms? This is the question. Which of these habitats has the lowest fraction of culturable microorganisms? Option A, seawater. Option B, freshwater. Option C, mesotrophic lake. Option D, activated sludge. Option E, soil. So here are the questions. Here is the first question and these are the options. Go through the question and post your answer in the chat box. And this is also the instruction for people who will be watching this recorded lecture. Post your answers in the comment box as we move ahead. Question number 1, answer A, B, C, D, any one you think. And then I will reveal what is the correct answer in a minute or so. Okay, I think it's time to check the answers in the chat box. Okay, I don't see any answer, but that's fine. Now let me reveal what is the correct answer to this question. Which of these habitats has the lowest fraction of culturable microorganisms? And the correct answer to this question is option A, seawater. Seawater has the lowest fraction of culturable microorganisms. Now before coming to the answer, Let us understand this concept of the great plate count anomaly. Now, what is the great plate count anomaly? So, this anomaly says that plate counts of cells obtained by cultivations are usually much lower by orders of magnitude than those from direct cell counts under a microscope. This also says that more than 99% of all organisms, all microbes from any environment, are unculturable. This is the great plate count anomaly. So, I think there is some noise in the class. So, I would request all the students to please turn off your video and mic, and you can communicate with me through the comment box. Please post your answers and your doubts in the comment box. Okay? This is the general instruction to everyone. So, yeah, we were talking about the great plate count anomaly. So, if you take a random uh, water sample or a soil sample and put it on an agar plate or any other media which can allow laboratory growth conditions, then only a few kinds of cells can grow on the plate. This is known as the great plate count anomaly because there are many reasons, many possible reasons, differing nutritional requirements. The organism may enter a non cultivable stage or a dormancy, and the organism may rely on interactions, thus cannot be grown in isolation. So that is the same reason why the answer is seawater, because factors such as failure of laboratory conditions to replicate the microbe's complex natural growth requirements, extremely slow growth rates, and presence of specific symbiotic interactions. This this these reasons are the main cause why many of the seawater organisms are not cultivable, because inside the sea there is high pressure and low temperatures in the deep sea, which cannot be replicated in the laboratory settings. So the great plate count anomaly refers to the fact that only a small fraction of microbes are actually cultured in the lab. In seawater, extremely low fraction, less than 1%, because most marine microbes are oligotrophic and require specialized conditions like high pressure and low temperature. Freshwater mesotrophic lake, slightly higher diversity of microbes than seawater, but still it is low. Activated sludge, this is enriched with nutrients, so more microbes can be cultured. Soil, more diverse and culturable microbes compared to the seawater. Thus, seawater has the lowest fraction of culturable microorganisms, and this concept is brought to you by Great Plate Count anomaly. I hope it is clear to the students, the concepts and the answer for the first question. If yes, then let me know in the chat box with a yes or no, and we will move ahead to the next question. Okay, I think we should move ahead to the next question. Question number 2: which of the following genes can be used as a functional marker? Option A, AMO-A. Option B, RPOB. Option C, 18S rRNA. Option D, 16S rRNA. Option E, ITS sequence. Understood the question? Which of these following genes can be used as a functional marker? Let's take some time and post your answers in the chat box as well as in the comment box, and I will reveal what is the correct answer to this question in a minute. Okay, I think it's enough time for this question. Time to check the comment box for some answers. Okay. Which of the following genes can be used as a functional marker? And the correct answer to this question is option A, AMO-A. AMO-A stands for ammonium monooxygenase A, which is a gene which can be used as a functional marker. Right? Now let us understand what is MOA and what is its function and how can it be used as a functional marker. So this is a schematic, simple schematic showing nitrification and related nitrogen transformation pathways carried out by nitrifying microorganisms or nitrifying bacteria. So ammonium is the readily available ammonium or ammonia is a readily available substrate on which the ammonium monooxygenase complex enzymes work upon. So it consists of three genes, AMO-C, AMO-A and B. This constitute ammonium monooxygenase. Ammonium monooxygenase oxidizes ammonium to ammonium hydroxide. Then comes the role of another enzyme, hydroxylamine oxidoreductase or HAO. How? HAO oxidizes further, sorry, HAO oxidizes ammonium hydroxide further into nitrite, which is then acted upon by nitrite oxidoreductase NOx to nitrates, which can be easily assimilated by plants, or nitrate can undergo denitrification. Nitrite can also undergo denitrification, and ammonium hydroxide can also have other fates, like nitrate reductase can convert it into nitric oxide, and nitric oxide reductase to nitrous oxide. Right, so ammonium monooxygenase is the first enzyme to act upon in conversion of ammonia to nitrate or nitrate assimilation by bacteria. Now there are two types of markers that are mentioned in the options. One is a phylogenetic marker or conserved genes such as 16S rRNA, 18S rRNA and ITS region RPOB. RPOB is a ribosomal protein. ITS region is a region between the 16S rRNA gene locus and the 23S rRNA. 18S rRNA is also another ribosomal gene locus. Now what is the purpose of such markers? These markers can tell us what is the community composition and taxonomy of a given sample. Exactly asking the question, who is there, the identity, diversity and evolutionary relationships between the organisms. Whereas the functional markers or metabolic genes will act as the functional markers, which will tell us that what is the functional ecology or what is the community function that is carried out by these enzymes or genes, such as ammonia oxidation by ammonia monooxygenase, nitrate K and NIRK and NIRS for denitrification, methanotrophy, PMOA and DSRAB for sulfate reduction. So these are examples of genes which act as metabolic markers or functional markers, which actually tell us what is the purpose of this community as a whole and what is the mineral conversion rate of this community. So such markers or functional markers or metabolic genes tell us what are they doing or specific metabolic capabilities of the community. So down here we use 16S rRNA, 18S rRNA and ITS amplicon sequencing to identify and the taxonomy of the community, to differentiate between species of the community, while metabolic genes and proteomics will tell us what are the functional ecology or what is the function of the community as a whole. That is why AMO-A or involved in ammonium oxidation, which is ammonium monooxygenase, is the correct answer, which acts as a molecular marker. I hope it is clear to the students. Let me know in the chat box. and we can move ahead with the question. Very good. Moving on to the third question. 16S rRNA based analysis is usually performed using total DNA instead of total RNA because of practical limitations. Assuming total RNA can be used for this analysis, what additional information would be obtained? So, did you understand the question? 16S rRNA is based on DNA sequencing analysis based on DNA analysis rather than RNA. But if there was a chance that RNA was possible, 16S rRNA was analyzed, what additional information could it give us? Option A, taxonomic identity of all organisms. Option B, taxonomic identity of all prokaryotic microorganisms. Option C, taxonomic identity of all metabolically active prokaryotic microorganisms. Option D, functional capability of all metabolically active prokaryotic microorganisms. And option E, total metabolic capability of prokaryotic microbial community. The question is up here. There are the options that I read out. Please take a minute or so to post your answers in the chat and the comment box, which will be followed shortly, and then I'll reveal what is the correct answer to this question. Okay, I think it's time for this question. Let us see if there are any answers in the chat box. Okay, the question number 3, 16S rRNA based analysis is usually performed using total DNA instead of total RNA. Because of practical limitations, assuming total RNA can be used for this analysis, what additional information would be obtained? The correct answer to this question is option C, taxonomic identity of all metabolically active prokaryotic microorganisms. This is the additional information we will get if we try a 16S rRNA analysis on the mRNA of the community. Okay? Now let us understand what is the difference between performing the 16S on DNA and performing the 16S on RNA. So in a metagenomics study, we collect the sample first, which is from seawater or soil sample or a contaminated area, extract the genomic DNA of all the microbes present, irrespective of their identity, and then subject this can be used, extract the genomic DNA can be used for two processes. One for amplicon metagenomic sequencing, where we PCR amplify and sequence the 16S rRNA gene, which is present in all bacteria, to identify what different kinds of species are present in the community. On the other hand, we can also do whole shotgun metagenomic sequencing, fragment the DNA and sequence whole genomes to predict the gene content, what are the genes that are expressed, what are the genes that are present in this community. It can also help us to find out, to aid us in the 16S rRNA gene based identification of the phylogenetic analysis. This can help us answer two major questions: who is there in the community and what are they doing as a community. Two major questions every metagenomic analysis should be able to answer. So DNA based 16S will answer us who is present, or the total community, dead plus dormant plus active. Doesn't matter what is the state of the microbe, since we are unbiasedly extracting the whole genomic DNA of the microbe, of every microbe present in the tube. So we are unbiased towards live, dead, dormant and active microbes. Every microbe's genome gets captured in the metagenomic analysis. Whereas RNA based 16S would say us who is active, only metabolically active fraction out of the total sample will produce RNA or mRNA, which can be used for 16S rRNA analysis, which will tell us which are the metabolically active species of bacteria that are interacting together. Thus, DNA based studies can tell us, analysis can tell us who is present, whereas RNA based analysis can tell us who is active and what function are they doing. Now, what 16S rRNA based analysis does? The 16S rRNA gene is a part of the ribosomal RNA operon, highly conserved across prokaryotes. Scientists sequence this gene to figure out who is there. This gives us the taxonomic identity by sequencing the 16S rRNA gene. Normally this is done with DNA because DNA is easier to extract and is also very stable. Sorry. Now, what is the problem with DNA based 16S rRNA analysis? DNA persists even after a cell dies, very stable. So when you analyze DNA, you are seeing active cells, cells which are dormant and also dead cells. This means DNA based surveys may overestimate community members. What happens if you use RNA instead of DNA, which was the question? RNA, the 16S rRNA molecules, are only present in living and metabolically active fraction of the bacteria. So cells making proteins and ribosomes, and ribosomes are made of RNA. Dead or dormant cells have little or no detectable rRNA. So, if you analyze total RNA instead of DNA, you get the taxonomic identity of only metabolically active population of the prokaryotes. So, not just every organism live, dead, or dormant, but only the metabolically active population you can capture when we perform rRNA 16S rRNA analysis on the RNA instead of DNA. I hope everyone is clear on this. If yes, then let me know in the chat box, and we will move ahead with the next question. Thank you. Moving on to the next question, question number 4. The term GC clamp is associated with which of the following techniques? This is the question. The term GC clamp is associated with which of the following techniques? Option A, ARDRA. Option B, DGGE. Option C, FISH. Option D, MLST. Option E, clone library. If you remember, most of these lecture week 7 topics covered these techniques. Now, where do you find GC clamp in these techniques? Which of these techniques has a GC clamp? Do not worry, even if your answer is wrong, you don't have to worry. I will tell you what is the correct answer and you will understand the concept behind it. GC clamp. I think it's time to check on this question, and let me check the comments. Okay, Ashwini has answered option B. The term GC clamp is associated with which of the following techniques? And the correct answer is option B. Correct, Ashwini, you have answered correctly. DGGE is where we use the GC clamp. Now let us understand these techniques one by one, because I have some related questions in the next slides from these techniques. Now what is ARDRA? ARDRA stands for Amplified Ribosomal DNA Restriction Analysis. Big name, but it's pretty simple to understand. This is a molecular technique that uses PCR amplification of the 16S ribosomal DNA followed by digestion with restriction enzymes and gel electrophoresis to analyze microbial diversity and structure by creating unique DNA fingerprints. So 16S rRNA, but the DNA sequence which codes for this RNA is targeted. It is digested with restriction enzymes. Now different species must have different sites for the restriction enzymes, creating unique size of bands of DNA cuts, thus known as DNA fingerprints. So what is the procedure followed in ARDRA? Microbial community from a contaminated site, isolate the ribosomal DNA and digest with restriction enzymes such as A or B, for example. Then suppose there are 4 different microbes present in this community. Microbe 1 has 2 restriction sites, A and B, like this. Microbe 2 has A, B, 2 sides of A and 1 sides of B. Microbe 3 has B and A oppositely, and microbe 4 has 2B and 1A site. So when the same locus of 16S rRNA is digested with the mixture of restriction enzymes and run on gel, each individual well will give us a unique fingerprint or DNA fragment corresponding to a particular 16S rRNA gene or the microbial species, which can then be compared with the reference database and genome for microbial diversity and genetic identity calculation. So this is called ARDRA, or Amplified Ribosomal DNA Restriction Analysis, done on ribosomal DNA. Now the other technique that is mentioned in the options is MLST, or multi-locus sequence typing. Now what is this technique? This technique is for microbes involving sequencing internal fragments of several housekeeping genes to identify their unique DNA sequences or alleles. Okay, so like ARDRA, which actually sequences 16S rRNA DNA, multi-locus sequence typing takes 3 or 4 housekeeping genes found in bacteria or in the microbial species, and then involves sequencing internal fragments to identify their unique DNA sequences. The unique combination of alleles for each isolate defines its specific sequence type. So let's say, for an example, locus A, locus B and locus C. If there is a known allele at all these 3, then they get a particular sequence type. So against the collection of publicly available MLST sequences, reads are binned on the most abundant closest reference for downstream analysis. Using majority rule on the aligned read, the consensus sequences are found out that in a particular housekeeping gene such as GAPDH or beta actin or Hox genes, if present in bacteria or in any other microbial community, then what are the different kinds of housekeeping gene change in sequences that can be identified in the housekeeping genes, since the housekeeping locus is mostly conserved, and such alleles are assigned to different locus. For each locus, the closest reference sequence acts as scaffold, and sequences are used to define a MLST sequence type. If SNPs are found, a new ST will be reported. More samples can be analyzed together, tracking recurring ST. So MLST depends on multiple housekeeping genes sequencing. ARDRA involves ribosomal rDNA sequencing, while DGGE, which was the answer of this question, is denaturing gradient gel electrophoresis, where GC clamp is used. So, what is this DGGE technique? Let us understand. Samples from different microbial communities are taken. The DNA is extracted, just like in any metagenomic approach, from each community. Add a GC clamp. This is an important step which anchors the DNA together once it's denatured. Right? So we will use a denaturing gradient gel, but the GC clamp will help the DNA to stay together even when it is denatured. Now what is a GC clamp? GC clamp is a short stretch of GC rich nucleotides, usually 40 base pairs long, added to one end of a PCR amplified fragment. So once the fragment DNA is extracted, fragmented, and then amplified, the primers can contain a GC rich region which can act as a clamp. And how? Because GC pairs form strong hydrogen bonds, like 3 bonds versus 2 in adenosine and thymine, making the fragment more resistant to complete denaturation. So in a sense, most of the sequence will be denatured, but the GC rich region will be difficult to denature. And why it is used in the DGGE process? In DGGE, DNA fragments are separated based on their melting behavior in a chemical gradient, like in the presence of urea. That is why they are known as denaturing gradient gel electrophoresis. Without a GC clamp, the fragment might fully denature and fall apart during electrophoresis. And the GC clamp ensures that at least a part of the fragment remains double stranded, so differences in melting domains can be detected. So based on the difference between melting domain, which will also be dictated by the sequence of the microorganism, these microorganisms can be sorted and taxonomic identity identified using a denaturing chemical gradient. So now if you run the GC clamp added PCR DNA fragment in each lanes, you can identify what are the shared genes among most community and what are the unique genes within communities which are not present in other communities in a denaturing gradient gel. I hope it is clear to the students that the GC clamp is associated with DGGE technique, denaturing gradient gel electrophoresis. Very
Yes.
good. Let us move ahead to the next question. Question number 5. Comparison of restriction site locations within the same target gene is performed during dash. So related question to all these techniques. Read the question carefully. Comparison of restriction site locations within the same target gene is performed during which procedure? Option A, ARDRA. Option B, DGGE. Option C, FISH. Option D, MLST. Option E, clone library. Let's take a minute and collect some answers in the chat and the comment box. Okay, I think it's enough time for this question. This one is a related question. So if you have followed the techniques closely, then you can answer this question. Let me check the comment box for any answers. Option A for Ashwini. Let us see what is the correct answer to this question. Comparison of restriction site locations within the same gene is performed during option A, ARDRA. Very good, Ashwini. This is the this is the technique in which we just learned that the 16S rDNA locus is tested with combination of restriction sites to identify different organisms present in the community. So coming back to the same picture, the ribosomal DNA is first isolated from the sample and treated with a restriction digestion mixture of one or two enzymes. Now if there are 4 different bacteria in the community, then they will have different DNA fingerprints or different restriction sites at positions which are unique in the 16S rDNA gene. And when such positions are cut by restriction sites and the product is run on a gel, they will give rise to different DNA lengths, bands of different DNA lengths, and thus forming a DNA fingerprint. So what does ARDRA involves? PCR amplification of the conserved gene 16S rRNA, digestion of the amplified DNA with restriction enzymes, and electrophoresis to analyze fragment patterns. Different microbial species have different restriction site locations in the same target gene. Comparing these digestion patterns allows differentiation of organisms. This is in the ARDRA. Now ARDRA, what is the purpose and advantages of ARDRA and when you should use it? Amplified ribosomal DNA restriction analysis, restriction digestion patterns of the amplified 16S rRNA gene. We have understood it nicely. What is the purpose of ARDRA? To analyze microbial diversity in environmental samples and to differentiate between closely related strains without full sequencing. So if the restriction digestion gives you different DNA fingerprinting, then you can easily identify one species from the another. To provide rapid low cost alternative to sequencing and often used as a screening tool, and after the screening you can go ahead with the top hits for sequencing. Now what are the advantages of ARDRA? This is simple and inexpensive, helps in rapid screening. This is also culture independent because you can directly isolate the 16S rRNA DNA and amplify it in PCR without having to culture it in laboratory. The sensitivity is pretty high based on restriction site polymorphism and useful for microbial community analysis. So this is the purpose and advantages of ARDRA, which uses the 16S rRNA gene locus for restriction digestion and obtaining unique DNA fingerprints. I hope it is clear to the students. Just let me know in the chat box and we will move ahead with the next question. Okay, thank you Moon, Bliss and Ashwini. Moving on to the next question. Question number 6. Which of the following statements about DNA sequencing methods is false? Okay, now we are talking about DNA sequencing. Which of these statements about sequencing the DNA methods is false? Option A. Sanger sequence is still a preferred strategy to sequence short DNA fragments at small scales. Option B. Second generation sequencing offers high throughput short read sequencing. Option C. Third generation sequencing offers high throughput long read sequencing. Option D. Third generation sequencing has higher accuracy than second generation sequencing. And option E. The Human Genome Project used first generation sequencing. These are the 5 options and you have to tell me which of these options is false about DNA sequencing methods. Let us take some time and collect answers in the chat and the comment box. I will revisit this question in a minute and reveal what is the correct answer. Okay, I think it's enough time and time to check the comments for answers. Ashwini and Moonbliss both have answered option D. Let us see what is the correct answer to this question. Which of the following statements about DNA sequencing methods is false? Correct answer is option D. Both of you guys are correct. Very good. Third generation sequencing has higher accuracy than the second generation. This is a false statement. Now let us understand the history of DNA sequencing, where it all began and what is the current status. So the DNA structure was solved by Rosalind Franklin and James Watson and Francis Crick, where Maurice Wilkins also showed one of the DNA images from Franklin. So, and then they got the idea of the double helix structure of the DNA. Then in early 1965, Sanger published two papers laying groundwork for DNA sequencing methods, and the first generation sequencing began, which is widely known as Sanger sequencing. Sanger refines the method for DNA sequencing. Applied Biosystems commercialized the Sanger sequencers. Then comes the second gen sequencing like pyro sequencing and high throughput sequencers and Illumina. And now is the correct status, third generation sequencing, long read sequencers, PacBio and nanopore handheld third gen sequencing. This is the timeline or history of DNA sequencing, where it started and where is it right now. Now, doing a comparison between the first, second and third generation, these are some of the important points. Now here I have mentioned the steps in which the sequencing was done. First generation sequencing or chain termination, popularly known as the Sanger method. First the DNA is purified, DNA is fragmented and cloned into vectors, which is then amplified by the bacteria, cloned into vectors, multiplied in the bacteria, amplified even more by the PCR, and then the sequencing begins. So dideoxyribonucleotide were used, which used to terminate these short sequences at random lengths. So sequencing by synthesis, read length was 300 to 1000 nucleotides, Accuracy though is greater than 99%. Now whatever is the last terminal nucleotide gets known via detection of these tagged DDNTPs, and such fragments are then mapped back to form the total DNA. There is no real time analysis, and the output data is very low output, it is not very high throughput. Second generation of sequencing, which is the Illumina sequencing, massively parallel sequencing, DNA is purified, fragmented, and then there is use of adapters at the ends of the DNA. Then what? Then a unique technique is used, which is known as bridge amplification. The adapters are adapters are polymerized onto a flow cell board, and then each end of the adapter is then amplified, and the free ends are tagged onto the plate via a bridge amplification. And then the nucleotides are tagged with fluorescence and detected, which detects the real time, which detects the sequence of the DNA. So this is also a sequencing by sequence synthesis, and the read length is 336 to 600 nucleotides. Accuracy greater than 99. Real time analysis no, but output data is high output. Now coming to the nanopore sequencing or single molecule sequencing. This also follows adapter ligation, but now this adapter is passed on to through a nanopore flow cell with the helicase activity, where each nucleotide is read. Single molecule sequencing read length 200 to 2 million nucleotides, so the read length vastly increased. Accuracy decreased 90 to 95%. It is in real time analysis, reading every base sequence and sending the signal, and the output data is also high output. So these are the key differences. First gen sequencing, Sanger sequencing, very accurate, long reads but too slow or expensive. Second generation NGS, high throughput, cheap but short reads. Third generation sequencing, single molecule, long reads, great for assemblies, but still needs to improve in the accuracy and also the cost of sequencing. Therefore, by far, if we take just accuracy parameter into account, second gen sequencing has the highest accuracy. Second gen and the first gen, not the third gen sequencing. So that is why option D was the correct answer, which you guys answered very correctly. Is it clear to the students? Just let me know in the chat box and we will move ahead. Okay. Now the next question, question number 7. Which of the following cannot be detected using clone library preparation? Which of the following cannot be detected using clone library preparation? Option A, pathogens. Option B, rare biosphere. Option C, antibiotic resistant microorganisms. Option D, eukaryotes. Option E, sulfate reducers. I'm sorry. Yeah, I think Okay, I think it's enough time for this question, and let's refer to some answers. Option B by both of the students. Which of the following cannot be detected using clone library preparation? Correct answer is option B, rare biosphere animals or organisms. Correct answer is option B, and Ashwini and Moon have both answered correctly. Very good, very good. Now, what are the steps in clone library preparation and what is a clone library? For any metagenomic analysis, we perform the clone library preparation, take all the genomes, fragment it, and put individual genomes into cloning vectors and let it grow inside a bacteria. Like, we collect the samples, environmental samples, extract the genomic DNA. The total DNA is extracted directly from the environmental samples. This includes DNA from both culturable and unculturable microorganisms. Fragmentation and cloning into vectors. Extracted DNA is fragmented, often using restriction digestion or mechanical shearing. So you use sonication as a mechanical method to shear the DNA into smaller pieces so that it can fit into the vectors. These DNA fragments are ligated into cloning vectors like plasmids. Vectors are cut with restriction enzymes to allow ligation of foreign DNA. So the restriction enzyme which digests the genomic DNA is the same restriction enzyme that is used to dissect the vector so that they have compatible sites and are able to ligate the genomic DNA fragments. Then transformation into host bacteria. Recombinant vectors are introduced into the host, commonly E. coli here, and formation. Each transformed bacterium carries a different environmental DNA fragment, and formation of a metagenomic library. A collection of recombinant clones is created. Together, these clones represents a library of environmental DNA fragments from the same type of environment. All the DNA fragments are cloned into individual bacteria, which is E. coli. Each clone can then be screened for either sequences or function. Sequence driven analysis like this, or function driven analysis for gene expression, mRNA level, protein level, proteomics study. Now, what is the rare biosphere? The rare biosphere refers to huge pool of low abundance microbial taxa present in microbial communities. Now, even though they occur in very low numbers, they can play critical ecological roles in the rare biosphere, such as resilience after the disturbance in the natural surroundings and hidden metabolic functions. How clone library captures the rare biosphere? The total community DNA is extracted from an environment which includes both dominant and rare microorganisms, and DNA fragments are ligated into plasmids. This is what we saw, and sequencing often of 16S rRNA genes is performed from randomly picked up clones, and clone libraries capture a less biased snapshot including some rare taxa. So, but the question was, which of the following cannot be detected using clone library? This is also a rare biosphere organism because the clone library will mostly contain abundant genome fragments of abundant species of organisms which may not be represented or underrepresented in a library kind of a setting, thereby missing the bias of capturing the rare biosphere. So these are one of the known limitations of metagenomic analysis, because the abundance is low. Many a times this becomes a limitation, in the clone library method, which is culture dependent, rather than using a culture independent metagenomic approach for looking for rare microorganisms. So metagenomic approach is better for rare biosphere organism rather than the clone library. So the clone library has limited sequencing depth, only few hundreds to thousand sequences. There is PCR bias because common taxa amplify more efficiently, random sampling limitations, and the cost is also more for representing the rare animals, rare microbes in the clone library. So this is the method of clone library preparation, and because of these reasons, because of low representation, because of PCR bias and limited sequencing depth of the clone library and culture dependent methods, the rare biosphere is not well captured in this method. Okay, I hope it is clear to the students, and we will move to the next question. Question number 8. OTUs are best described as dashed. What are OTUs? Best described as option A, groups of similar species, option B, groups of similar strains, option C, groups of identical cells, option D, groups of similar DNA sequences, option E, groups of identical DNA sequences. Some confusing options, and you have to tell me which is the correct answer for question 8. What are OTUs best described as? Let's take a minute or so and collect some answers. Okay, I think it's time enough for this question. OTUs are best described as dashed. Let us check the comments for answers. Ashwini says D and Moonbliss says D. Correct answer to this question is option D. You guys are correct again. Groups of similar DNA sequences are known as OTU. Now, what does OTU stands for? OTU stands for Operational Taxonomic Unit. It's a practical way microbiologists classify and compare microorganisms in environmental sequencing studies. So grouping similar taxonomic units into a operational taxonomic unit, grouping some bacteria to classify better these microorganisms. Since many microbes cannot be cultured in the lab, OTUs provide a way to group organisms based on DNA sequence similarity. So this is the sole basis for formation of OTU. Based on DNA sequence similarity, many organisms are grouped together to form a taxa, since most of these cannot be cultured in the lab. Similarity threshold: 97% of sequence identity is used as the cutoff or a stringent threshold, 95% cutoff genus level, and 90% at the family level. Now, what is the purpose of operational taxonomic unit? To estimate what is the microbial diversity present in the sample. In fact, checking how many different OTUs are present in a sample, and to compare microbial community composition across different samples. And why not just species? Many microbes haven't been formally named or cultured, and DNA based OTUs let researchers study them without needing traditional taxonomy. For example, if you sequence the 16S rRNA genes from soil and cluster them, all sequences greater than 97% grouped into the same OTU, and each OTU represents a putative species. Though in each OTU there can be different species, but since many of the organisms are yet to be discovered and named, we can treat them as a single taxonomic unit and move ahead with the analysis for studying the greater microbial diversity and community composition. So looking at this schematic will help us understand OTUs better. So when we have a bulk sample, we do PCR. There are many species that we identify. The big circles represent here species 1, 2, 3, 4 and species 5. Now in the amplified DNA or amplicons, we have some of the clusters separating with PCR errors. There are some chimeras and primer bias, some species lost due to insufficient sequence depth. Then bioinformatic clustering of these sequences, removing sequencing errors and tag jumps, gives rise to what is known as OTUs or operational taxonomic units, which tell us that one OTU can be considered as a single species in further analysis due to more than 97% of sequence similarity. I hope it is clear to the students what is an OTU and how is it used in the metagenomic and bioinformatic analysis.
Yes, sir.
Good, thank you. Moving on to the next question, question number 9. The lowest taxonomic level till which we can usually reliably identify a microorganism using 16S rRNA amplicon sequencing is dashed. To which level can you identify correctly in a sample just by using 16S rRNA amplicon sequencing? Option A, variant. Option B, strain. Option C, species. Option D, genus. And option E, family. Let's take a minute or so and collect some answers in the chat box. Okay, I think it's time to check some answers. Ashwini says D and Moonless says B. Okay. So the answer to question number 9, lowest taxonomic level till which we can usually reliably identify a microorganism using just 16S rRNA amplicon sequencing is genus level option D. Option D is the correct answer, so Ashwini is correct. We can identify the microorganism certainly and reliably till the genus level. Now again coming back to the 16S rRNA, which is a boon to metagenomics and identifying the microbial communities. It is widely used for identification because it is conserved but also has variable regions. However, its resolution is limited. It cannot reliably distinguish between strains or very closely related species, such as E. coli versus Shigella. It is most reliable at the genus level. Sometimes species level assignment is possible if the sequence is very distinctive, but not always as a reliable across all taxa. So what we do is we identify the 16S rRNA, isolate it from the bacterial ribosomal uh, proteins, then PCR amplify the 16S rRNA gene and sequence it to identify the genus level of the bacteria like lactobacillus. So we can only go reliably till the genus level. Sometimes species level information also can be obtained from the 16S rRNA sequencing, but not always. Only if the sequence is very distinct, we can tell the species apart. Okay, so this was a short explanation. I hope it is clear. And then we come to the last question of this session, question number 10. Assume that ABC1 gene is a conserved gene responsible for antibiotic resistance in bacteria. An NGS based amplicon sequencing experiment is designed where both 16S rRNA and ABC1 gene are targeted in separate runs. When the data is compared and interpreted, which of the following is not likely an outcome? So you understood the question? ABC1 is responsible for antibiotic resistance. Then you did 16S rRNA sequencing and also ABC1 gene based amplicon sequencing, and then compare the data. Which of the following is not likely an outcome? Option A, 16S rRNA based analysis will give more OTUs than ABC1 based analysis. 16S rRNA based analysis will reveal taxonomic composition of the microbial community. ABC1 based analysis will reveal abundance of antibiotic resistance gene carriers. Option D, this study cannot determine which microorganisms are carrying the ABC1 gene. And option E, this study can determine if ABC1 gene is horizontally transferred. Understood the question? Read the question carefully and tell me what is the correct answer to this question. Okay, I think this is enough time for this question, though it is a little bit complex, but we will be able to understand this question better once I reveal the correct answer. Let me check the chat box for answers. Moon Bliss says option C, okay, and Ashwini says option E. Let us see what is the correct answer to this question of this designed experiment. Correct answer is option E. Horizontal transfer cannot be studied by 16S rRNA metagenomics and ABC1 targeted sequencing. So, option E is not a likely outcome of this experiment. So, Ashwini is right again, and Moon can try in the next class for some correct answers. Let us try to understand what this experiment tells us about. So again, coming back to two different types of questions that can be addressed in a microbial community. 16S rRNA sequencing answers the question, who is there? Very important, who is there? Taxonomy or community composition of the sample. While functional marker analysis, like we saw in the ammonium monooxygenase question, ABC1 is an antibiotic resistance gene, so this is a functional marker analysis which tells us about what can they do or functional potential of the community. Now the marker type, universal gene which is present in all prokaryotes, but functional gene which is antibiotic resistant gene present only in some organisms. Now what is the diversity it captures? Broad, the diversity is broad and many OTUs can be covered in this 16S rRNA. That solves some of the options. But for functional gene, the organisms carrying the target gene are narrow, so it is narrow spectrum. So ABC1 will reveal actually the abundance of these gene carriers, and rRNA based analysis will give us more OTUs and also reveal the taxonomic composition. Information obtained, taxonomic identity usually up to the genus level, and functional capability like antibiotic resistance or nitrogen fixation in case of AMOA. Limitation, it cannot tell us about the functional traits and it cannot tell about the full taxonomic content, who carries the gene. So this also rules out option number D. The study cannot determine which microbe carries the ABC1 gene. Of course it cannot. It can only say that there is an relative abundance of antibiotic resistance gene carriers. Now 16S rRNA gene is again a taxonomic marker for who is there, and ABC1 is which antibiotic resistance genes are present. We need, and looking for each option, 16S rRNA is highly diverse, giving many OTUs. Functional markers are less diverse. 16S rRNA will reveal taxonomic composition. ABC1 based analysis will reveal abundance of gene carriers. This is partially true. It will show the abundance but not exactly which taxa carried it without linking. Cannot determine which microbes carry it. This is also true. And horizontal transfer cannot be detected by amplicon sequencing. We need the genome context and the plasmids and the mobile elements like transposons and not just the metagenomic gene counts. This is why option E is the correct answer. It is most likely not an outcome of this experiment. I hope it is clear to the students. And with this, we have come to the end of the presentation. I will stop my screen sharing. Yeah. So with this, we have come to the end of the presentation. We have solved 10 different questions corresponding to lecture week 6, where we dealt about techniques, where we also saw the comparison between the functional markers and the taxon identity markers and the genomic markers. I hope you all liked the class. This class was informative and you could clear some concepts. If you have If you are satisfied with this class, then well and good. If you have any more questions, you can ask me, or I will end this call. The recordings of this class will be uploaded on YouTube very soon, and the materials, the slides that I have made for you guys, will also be shared to you by the NPTEL authorities.
So actually, I wanted to ask something in regard to the examination.
Okay, yeah, please ask me. If I have any knowledge, I will
So,
help you out. Yeah.
so like I'm in the remote area of Nagaland, so is it possible for me to do exam, write the exam? Like I have applied for the three courses at the same time,
Okay.
so there won't be a clash of schedule, right?
No, no, no. I don't think so. So you will have different times for all the examinations. I don't think there will be a clash between two different examinations. While
Okay, sir.
I exactly do not know when is the environmental biotechnology exam going to happen. That will all depend on Professor Pinaki and the NPTEL authorities. But I assure you that the exams will happen at different times. No two exams will clash.
Okay.
Yeah. Okay, thank you for joining, and I will end this call. See you next Saturday with lecture week 8 questions. More 10 more or 15 more questions for you guys. And yeah, happy weekend. Enjoy your weekend, and I'll end this call. Thank you for joining.
Thank you.